Hiya. Yeah, so the feed dropped for a little bit. Apologies for that, but we're back on right now. So
So, hello again.
hi again. Joan and I kind of continued rambling on, thinking we were speaking to you guys, but I think that's one thing on a boat. It's so hard. Obviously, it's already amazing that we have such great connection and like we
Yeah.
can live stream from such a remote place. So it does come with obviously sometimes we get problems with our connection, but it doesn't happen too often, I think,
No,
considering.
the internet is much more stable than I expected.
Oh, yeah, same. I mean, I was almost expecting to have,
No internet.
barely any,
Yeah.
but, we're still allowed to contact our families and
Yeah.
watch movies at night. Well, not really streaming, but, we still we have quite a lot of flexibility to access
Definitely.
things, especially if we're working on, our projects or anything. We can actually still read papers online and yeah, so we're planning on, if it's possible to fit it in the quiver, yes please, yeah. So yeah, we're going to be collecting this black coral here for our genome sampling project, so we're keeping it in the quiver to flash freeze it in a
Yeah.
bit.
And until now, this branchy type, we still don't have a lot of samples to do a very detailed description of it. So when the rest of the sample, so part of it will be preserved in liquid nitrogen for genome project, and the rest will be preserved in ethanol, just for general identification. I think Jeremy would be really happy about it.
Yeah, exactly. So for these, we kind of have these small cryo valves that we cut off a tiny fragment with tissue on it, and we put it in, and we flash freeze that, so we don't actually, it's, just alone the fragment inside a cryo valve without any other things to store it in. And yeah, that very low temperature is what kind of preserves the tissue so that we can get as pure of a DNA sample as we can. And yeah, it's always amazing because we get to often get little critters on them, or brittle stars, which, as you were speaking, I don't know if the stream went down when Joan was speaking, but talking about that amazing relationship that we know so little about. I know with the brittle stars. I've read a lot of varying papers where some people think it's an opportunistic relationship because the brittle star is the one that's using the coral, but I've read others where you often see a coral that's half dead, half healthy, and you always get the brittle stars on the healthy regions, and you ask yourself, is it because they have a symbiotic relationship and the brittle star is actually helping the coral, or is it
Just use the
or are
coral,
they just using
like a
that
cold-blooded
part?
animal.
Yeah, I've read one hypothesis that because they climb the corals, so they can filter feed at a more advantageous position in the water column, this might actually help them catch things like, hydroid larvae so that they don't settle and kill the corals as easily. So that might be one reason how they have a symbiotic relationship. As we said, in deep waters like this, we don't we can't say anything 100% for sure. There's so many hypotheses for these kind of things. I'm sure our scientists on shore probably have other explanations. So it's one of those things that's just super fascinating. You could talk, I think, for a really long time about.
And just saw a lot of people commenting saying they missed the shark. So just recap that a little bit. It's a white-tip reef shark, so that is all right. It's not a very it's not an endangered species. So if you're a diver, you will be able to see them quite a lot. And for us, it also swam really, really fast. It's way faster than our camera, so we didn't really see it for a long time during that streaming break off. But hopefully we can see it later on.
Yeah, hopefully. We were actually concentrating on the coral just now.
Yeah.
And then Vicky pointed out that she saw a fin, or I think the tail, and we turned a little bit and there were two of them, but yeah. No, I think we've been quite lucky with sharks. I haven't really been expecting to see many, I think, this deep,
Exactly.
but last, I believe it's last dive, we saw those silver tips. Obviously there's the cat shark, that was something that I've never even heard of before, so...
And today we saw the mermaid purse. That's my first time seeing a live one with egg yolk in it. Yeah, Will just mentioned usually for a white-tip reef shark, it stays around usually less than 40 meters, but this time we saw it at 181 meter-ish depth. So basically 200 meters, so it's quite amazing depth that it can get.
Yeah, it's crazy to think about their range, how, you know,
Yeah.
you can see them, in 2 meter depth water, and
Exactly.
you can get them here as well.
I remember when I was in Malaysia, there's a dive site near Sipadan. It's just basically for white-tip reef sharks, and during the daytime they just lie on the slope, don't move at all.
Yeah, they're really cool creatures. It's a little bit sad though, I have to be honest. I think with both white tips and black tips, you know, they're, as a diver, they're almost quite common.
Yeah.
That you almost start to take them a little bit for granted, because you just see them everywhere. But then if you haven't seen them in a while, it kind of reminds you, oh, actually, you know, there's still a shark, there's still something amazing.
Yeah.
I haven't seen one in a while, so it's...
Yeah, me neither.
But also I haven't really been diving in a long time either.
Thanks for collecting the species. We will keep going on.
Yes. Yeah, so I'm still, I guess, still baffled by this reef that we found. The shape of it is just... I mean, I'm no geologist, so I think we can only really ask other people, you know, what... Do we have an idea how this formation came about? I don't even know how to begin to explain it. Does anyone have any hypotheses?
Definitely. I was listening to Rob's explanation
Yes.
yesterday.
Yeah, sure. I mean, it's certainly complicated, but you know, we're talking a 500-meter tall reef. They don't grow from the bottom up. It's more like the top down, and so if you imagine the ledge would have been much shallower, and the coral reef when it started to form would have been within very shallow waters, you know, photic zones, so coral reef could develop. And then what's happened is over long geological time, this ledge has actually been pushed downwards. It's called subsidence, and so it's subsided at least 500 meters down, but the living portion of the reef has continued to grow because it's remained within that sunlit zone. So I used the analogy before of like a kitchen table with a plate on it, and let's say you take a couple of inches off the bottom of the legs of the kitchen table, that's got a lower, but if you throw another plate on there, the relative height of the plate stays in place while the kitchen table just gets a bit lower each time. And what's happened is the ledge has just been has subsided down at these deeper depths, but the living portion of the reef, at the very shallow top part, has continued to grow, and so literally you've added one dinner plate on another, and these are these layers that we are seeing here now. So of course the deeper parts of the reef are older, the shallower parts are younger, and what we're hoping is as we get up onto the crest of this 500-meter tall reef, we will actually see living shallow water coral, you know, zooxanthellate shallow water coral. But there's certainly a lot more life appearing. We're at 170 meters, lots of little soft corals and sponges appearing, stylastrid corals. So yeah, you can explain it away from a you know, long geological time, but the living portion of it has always been at the very top.
And as you mentioned, Rob, you can already see that it's already becoming way more colorful with the coralline algae, and you know, that's almost a funny distinction you that kind of demarks the mesophotic zone that you get so drastically, and now you can really see that. Oh, what is that little? It's it seems like a black coral. We are collecting a rock sample right now, but it seems like a small black coral in the bag. Maybe it's very thin, but yes, maybe after obviously collecting this, we could have a zoom in and take a look at it.
That's just crumble. This little
Looks like really hard rock here.
Yeah. Actually, throughout this dive, I feel like we've been seeing quite a lot of hard rock, whereas on other dives, there's been a lot more crumbly substance. Yeah. No huge slab of rock today that we have to nudge between two bio boxes, which is good, or maybe bad, I guess, for some people. Yeah, the reason why Joan and I pointed out the, well, we think it looks like a black coral in the back. You can see that tiny little strip is because one of our target corals is a Bathypathes, which is very, very small, very fine, which, you know, we've been looking for, but unfortunately, it's because it's so fine and small, you really have to be so close to the substrate to be able to notice it, and you often have to be at the bottom and
It's often like on a sandy bottom, which would tend to not to disturb the bottom sediment.
Yes, exactly. And Jeremy says, go ahead and try to say the species name. Bathypathes, correct me if I'm wrong. Yeah, it's one of those.
Just say it fast and no one will know.
Yeah, I'll just go blah 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 blah. Yeah.
And this is a species name that when I put it into Google, no pictures will show
Oh
up.
no, exactly. I think the only reference we had was a picture from the last trip that Jeremy was on, and he sent us that one picture, and we've been trying to look for it, but because it's, as we said, so fine, you know, when we're skimming over the sand, we kind of can feel like we see it everywhere, but also at the same time, it's hard to, you know, ask the pilots to zoom in on every single thing that we think looks like a tiny branch. So it's...
It's like a stack of shell.
Yeah. Alright, moving on. Alright, and
Yes.
okay.
No, that doesn't really look like the these branches don't look as long.
No. There's a little fish in the background just wiggling.
Oh yeah, a very small one. Yeah, we've seen quite a lot of fish, I think, this dive, especially on our way down, getting all the hatchet fish. There were plenty of them. And a lot of them almost seemed to be swimming down with Sebastian during our descent. I was trying to keep up.
Also another rainbow runner. We're
Yeah.
staying with our like back angle camera for a really long time as we're going down. Sometimes you just feel like these fish are trying to just stay in front of camera so that it can be famous. Now we're zooming into the coral,
Yeah, it does not look like what we were looking for, but
yeah, let's wait until it's focused.
Yeah.
It looks like a lip coral. know if Jeremy's with us right now.
Jody's keen on the
Yeah, we got one. Just keep an eye out as we got friends.
okay.
I've got 2 eyeballs in that. Monsters. What are they? Okay.
Is this the closest?
No, it's a move, yeah. I can move.
Sticopathes.
So
Okay.
Baptist, I think did we have
Baby?
This, yeah.
Baby Sticopathes.
We could move on.
You want it?
No.
No.
Thank you.
Now it's sticky outy rocks.
Indiatis?
Sticky outies.
I'm already looking forward to getting back to the lab and sampling everything we've got, especially with that coral that Cody was trying to get off the arm but was sticking onto it. And Joan and I have already had a few samples that once they reach the surface,
They
the
just
tissue
have-
almost looks like it. Okay,
It's
not the tissue, but like
melting.
there's this goo. It's melting
Yeah.
and it's gets sticky everywhere. A lot of the black
excrete
corals
a lot
have
of
that.
mucus.
Exactly. So exactly, making everyone hungry.
I mean, another word for mucusy is like they're quite salty. So still,
Snotty.
Well, I'm
Exactly.
trying to shift it to a more Dana language, but if you're
Don't we love a good old snotty coral? So I can already imagine that that sample is going to be a fun one to handle in the lab. Yeah. Especially in the first few minutes, I think, when we have to deal with the flash freezing samples.
yeah.
It's, I mean, I wouldn't say chaotic, but it's like an organized chaos in the lab where we have to rush, but, you know, obviously we can't
Still organized.
Still organized, that's why I say organized chaos.
I think we get a hold
Yeah,
on it, and yeah, the whole flow is quite smooth.
yeah, we've got
Yeah,
a,
with everybody, literally everyone on the boat is helping us.
Exactly.
The ROV team is taking off the samples even before
exactly.
everything.
Yeah,
being
Marty's
strapped down.
Marty's often there to spray our other samples with ethanol while
Yeah.
we take care of the flash freezing samples.
And Vicky will help to just move around, give us a hand at any time.
Exactly. So everyone's always super helpful. And then after the flash freezing, we usually can, you know,
Got
take our
rock.
time with everything else.
Find that for us.
Go ahead and 2.
Yes, please.
So as we come to our penultimate dive, Joan, do you have one specific highlight to most of the dives that we've been doing?
Highlight.
Yes, I know there's been
There's so
plenty of
many,
highlights. It's hard to
exactly.
pick one, but
I would say still until now, even though the Dumble Octopus was
Yeah.
amazing.
Yes.
It's like definitely your highlight.
I think that was gonna be my highlight.
Somehow I just really like those teeny tiny stuff. So like today, the
Yes.
little worm on the sea cucumber. I think that would be my highlight today. And previously,
Oh yes,
Ooh,
the
and
rockometer.
today all the rock structures. Apparently, we're having the rockometer on screen.
We're up 5 now.
Yeah, 5. Our classic Falkor meters.
Maybe.
And at one moment, that was when I'm having lunch. I saw them grab a huge piece of rock, but eventually it broke. I
Oh.
thought we were going to have another
Avalanche.
situation like last
I
time,
know.
having a big rock in those
A situation.
We can do it next season.
Just create the biggest avalanche for our departure. Yeah, we did see that.
Yeah.
It's so tempting to keep those huge slabs of rock.
We are looking at might be octocoral.
Show it. Extra pot. It looks live.
Yeah, and a gastropod right next to it.
It's a beautiful one.
There are actually 2 corals here,
Yeah.
and some squat lobsters hiding in the caves.
Yeah, that's a
Pretty.
beautiful.
Yeah,
That is a cool shell. That's a beautiful
it
octocoral as
almost
well.
look like a baby, but the shape is different. But the pattern does look like trident a little bit.
Mm-hmm.
Yeah. I know that like at Ames, they're trying to breed trident since it's the natural predator of crown of thorns.
Ah, yes,
But
I did
so
see about
far,
that.
Yeah, so far it's really, really hard,
Yeah.
and I don't think we've heard any of successful cases
No,
yet.
I have heard that was a big project. Did you help on that project?
No, I was only observing.
Yeah, I did read about that, and I thought that was great. I mean, with the crown of thorns problems, it's something that needs another method of trying to eradicate them rather than just human efforts. I worked in Raja Ampat for a couple months, where one of the projects that I helped with was a crown of thorns eradication program. And we tried loads of different methods, but they were just so heavy on human power. We had a group of about 40 people going out to different sites with these huge wooden tongs, kind of just picking them up, and we dug this hole and we had to burn them. And there's been multiple ways that people have been trying to eradicate them, but burning seems to be one of the burning or injecting them, which costs a lot more. And in Raja Ampat, I think they preferred the burning method. I have heard of some people just trying to bury them in the ground without doing anything, but somehow they have found a way to get out of this hole and almost crawl their way back to the sea, which is scary to think about because they seem to be these almost subhuman, immortal things that can't die from.
They're really, really, really tough.
Yeah, they really are. So, yeah, crown of thorns. They're one of those animals that I think when I first started diving, I looked at and thought, wow, they're beautiful because, they have these bright blue colors. Yeah. They really do look quite nice, but once you take them out of the water, they
Smell so bad.
They smell so bad. They instantly, talking about slime, they morph to this, vicious-looking, slimy creature, and yeah, they're a very weird animal.
And I remember back when I was working, because it's a resort, and for sure, and there was an outbreak of crown of thorns in Maldives. They were saying they really don't want coral to be eaten by crown of thorns anymore after the bleaching. So they're like, we really need to pick them up. And here I really need to apologize to my ex-colleagues because when I picked them out of the water and I forgot them in the phone boxes back at the office and left them there for 2 days under 30-degree sun.
Oh, wow.
And yeah, the whole office
the
was
smell.
absolutely horrible.
Yeah, it's crazy how they smell, really. They smell like the worst fish market you've ever been to in your life. Yeah, actually it was similar to that time I worked in Raja Ampat, where I think over 2 days of working on this project to eradicate crown of thorns, we were only 30 of us. We managed to collect over 200, 300 crown of thorns and filled up a boat with them. And cleaning the boat afterwards was not nice. It was, as I said, slime everywhere, smelled awful.
Yeah.
But yeah, it was quite amazing to think that in just 2 days we could, you know, get 300 about of them.
And now we're trying to collect this species into our quiver. We're filling quite a bit of our quivers this dive.
Yes, last sampling dive, so we have to try and get as much work done as possible.
And hopefully, as we move on later, get into shallower areas, we can see the branchy one that Jeremy was looking for. It's really hard, but we will still try to keep our eyes wide open.
Yep. So, what is your mum planning on doing today for her birthday? Is she
Well-
watching the live stream or?
Fortunately, she couldn't. Yeah, but it was actually her birthday yesterday, and in the time back home, and she did a pretty good like. She and my dad made really good dinner, and they were sending me pictures.
Oh,
and
that's
just
very
like,
cute.
Yeah, they're making dumplings.
Oh,
And,
yes.
Oh, I really want to have some.
Oh, what a great family tradition to make dumplings. I do love that.
I think somehow for Chinese, especially from northern part, whenever there's an activity, dumplings will always be the thing come into our mind. Yeah, and it's easy to make, and it can be a family activity if everyone participates. Someone make the dough, someone wrap it up, and
Yeah.
someone cook it.
Yeah, exactly. So yeah, I'm also half Chinese, but my mother, who is the Hong Kong Chinese one, out of my parents, actually does not like dumplings. So
Yeah.
that was not really a tradition for me growing up. But I have tried to do it a couple times ever since leaving home, and yeah, it's just a fun activity
It is,
to do with people. It really is.
I would have had it that night.
Yes.
Yes,
please.
this one.
Every time I still feel when we zoom in, it looked like such a big colony, and now
Yeah.
it's like it is really tiny.
It's really hard to grasp the scale of things. It's only really once we get to the lab that, you know, I see it and I think, oh
Yeah.
wow, this is way bigger than I expected, or smaller, or like
And our pilot Chris here just called Cody our arm magician, so that he can grab anything that we want and put it into a quiver box.
oh yeah,
Now we have the magician J right.
oh yeah, J-Rod is on the arm.
J-Rod right now.
We're all well, all the pilots are magicians here.
Awesome.
Unfortunately.
I always get a little bit nervous when I watch this whole process.
Is this one also a little bit sticky?
Add to the slime.
This little crinoid came all together with the arm, and now it float towards one of our ball boxes, and now it's swimming down.
Actually, didn't really see it swim in quite the beautiful way that we've seen it swim before. I always love watching those when they kind of dance in front of the camera
Yeah.
for us. Those and flatworms, I'm always amazed by.
How they're flapping
Yeah.
underwater.
Actually, speaking of favorite marine animals, like flatworms and nudibranchs for a long time were some of mine.
Yeah. Especially if you like taking pictures underwater.
Exactly.
Yeah, they're
That's-
one of the best model in
Oh,
front
for
of your
sure.
lenses.
Exactly, for macro photography, it's especially because they don't move very fast and they're so different, they're so colorful for a macro photographer. You know, I love underwater photography. I could easily spend half an hour of a dive just sitting with a tiny nudibranch taking pictures of it.
Yeah.
You know, you could have a shark swimming by me and I'll concentrate on the nudibranch.
Anything else over here?
uh, Yeah, and I think especially as like a photography enthusiast, you know, seeing this 4K camera and not even having to be the one underwater to control buoyancy and all of that, it's made so easy for us. I guess we're not the pilots, but to just be able to sit back and relax and see all of this in such great quality. It's
We had a really good discussion on our YouTube channel right now. People are saying the most, the common, the fish's common name that is leaves them with the deepest impression, and there are quite a few good names.
sarcastic fringe head.
And there's a bony ear ass fish. I've never heard of it.
There was the Western Australia fish.
Yeah, I have heard of that.
One of your actors, yes. Did we catch one?
It's one of those that, you know, you get on those dive slates and you see a picture of and everyone points it out because of the funky name.
Yeah.
Yeah, we caught them. That's
And there's a fish's scientific name is bastard trumpeter.
Sadly, our live stream has to, you know, restrict some of these comments because they
Yeah.
think they might be foul words.
Yeah. Sorry, well, your comments couldn't really be shown. Thank you for all of our fish experts on shore to share all these interesting information with us.
Yeah. It's like it could
Can
be
we
a...
zoom in, please? Yeah. That looks like an octocoral. See how it looks when we zoom in?
Yeah.
Might not be.
Yeah, I think once we see the tentacles.
Yeah.
I'm thinking
Looks very
octocoral.
dusty, no?
Yeah. Might just be blurry from the focus. Oh.
it's really hard, but we can skip. But is that a nudie?
Oh. It's not moving though, so it's hard to tell.
It's too hard. We can skip this
No, I
one.
can't actually set up. I was trying to do two things at once.
Yeah, no worries.
I think this type of benthic substrates reminds me of all those muck diving we did before. Just it looks quite not alive, but usually if you pay
Yeah.
a lot of details just to look in between those rubbles and sand, it is actually very vivid in all terms.
I think those are actually some of my favorite dives. The ones that, yeah, you look down and it just looks like no rubble, but then you start to look closely and you get, the most wonderful little creatures hiding. And I think it's actually through mud diving that you get all the weird specimens.
Yeah.
Yes, this looks like dead.
Yeah.
It looks
I
very,
think we can skip
healthy.
on this one. It's alive still, but it's
yeah,
not
it's,
something.
oh yeah,
we're
you see it's an
interested in.
octocoral.
Yeah, but
See the
it's half that
tentacles?
maybe.
Okay, well, thanks anyway.
This is definitely another octocoral. It's really, really
So
colorful.
the white ones, they're stylastrid
Yeah,
hydrocorals,
the
or
stylastrid.
hydrozone.
Right now we're showing the inset from our. It's not from another angle of our camera.
Situation camera.
Yeah, you can see it's really a steep drop. It's quite scary almost for people with vertigo. Yeah. And yeah, as Chris said, it's so narrow. You can almost feel like you could hug it.
Is that a black coral there?
I can really see the current is picking up as well today.
Yeah.
Like the marine snow is moving so fast. Lots for the filter feeders to feed on.
I think for Gorgonians, they might really like this environment.
Yeah, exactly. And usually when you get into about this depth in the mesophotic zone, the previous dives we've seen, we've found gigantic Gorgonian sea fans, bigger than any I've ever seen in my life, and I feel like I've seen some pretty big ones.
And here we're looking at
Another,
a Hexa Coral.
I believe. Oh, I think it's more of a whip, so soft. Octo.
Jeremy is trying to contact us. Can we have a bit zoom into the polyp rows, please? It's a black coral.
Black coral.
Now we're trying to stabilize the ROV. This is really, really steep, and can
And
see
it's-
we have two arms, kind of arms. They're sticking out from the bottom of the bottle box, and every time it can help us to either stabilize the ROV, or sometime it can be used as an extension to expand the area for storage.
Yeah, and as Joe and Jeremy have said in the comments, it looks like a pseudocirripathes, which apparently would be the first for the entire country. So
That's exciting.
that would be, yeah, another first for Falkor and SuBastian.
We've got another fun name from Rare Art. We've got a moth name. It's called La Cerveza. It must be a very drunk moth. Yeah, we're trying to see if it has one messy row of polyps.
Often when we have to look at things that are so hard to focus on, we have to use the manual focus, so
Yeah.
that's why it takes a little bit of time to just adjust it.
We're getting there. The color is quite nice.
Yeah,
the
it's
tentacles
beautiful.
are having like a gradient color towards the mouth opening.
Yeah, it'd be nice to see if there's anything living on it as well. Okay, yeah, that's beautiful.
Yeah. Does this look like a messy row of polyps to you, Jeremy? I think it is a little
Kind
bit
of,
irregular.
Yeah, it looks like it's mainly on the one side.
Some of
So it
them
looks
are
like
like
it's-
alternating in the middle part here.
Yeah. But it does seem to be on the side that we're
Yeah.
facing rather than on the other one. So,
Let's collect this one.
yes. This is we can break it off so we don't take
Yeah.
the whole thing.
Going in a quiver?
Do you want it to go in a quiver, Jeremy? Bell box is fine.
Firebox, yeah.
Which bottle?
Do you think? I
Do we
think.
have space in one
I
of
think
the-
this is long one for the bathroom.
Yeah, 3, 4 maybe. I think we only have 2 other samples in there. Yeah, it's very interesting to see that just having one row actually.
Yeah.
You don't often see that on corals. Okay.
Not that much?
You can. Yeah, just break it off at the
yeah.
curve.
Not your fine.
Yeah, that should be good.
Jeremy gave us a warning saying this coral will be super hard to break.
As
So
we're currently witnessing.
yeah. Luckily we have all the magicians on boat.
Yes. So I guess bear with us as we try to collect this.
Yeah.
Might take a little bit of wiggling around. Yeah, I think what we need is a third arm somewhere to be able to come in, but just having scissors at the tip so you
Yeah.
can just cut things
There
really
we go.
easily. Nice, perfect. Yeah. Anything that, yeah. Sure. A quick look at the big polyps on it. Yeah, this is a black coral that I've never seen before.
Maybe not is the bottle brush.
You can see the tentacles are beautiful. They have this gradient color with
Yeah.
like the white at the tips, like the orange red at the base. And same actually with I think the whip itself. If you look at the base in the background, you can see it's
And
nice
it's a
and
black
white.
coral, so it actually having a black backbone
Yeah, exactly.
under the tissue.
I guess that's really why, it's so important to count the tentacles because, you would think that for a lot of black corals you can see the black skeleton, but in this case, you really can't tell that the skeleton is
Exactly.
black. So you really have to, count the tentacles as hard as it is sometimes. Okay, well. Yeah, I think that's my favorite coral sample of the day. I've never seen anything quite like it.
Somehow I think my favorite one's still the one with shark stacks on it.
Oh,
It's
yeah.
just,
That was
yeah, the coral itself doesn't look that pretty, but it's been chosen by the shark.
exactly. So it must be special. Yeah, I feel like we've really seen such a wide variety of black corals on this trip.
Now we will start moving up.
Yeah, to the top of the reef, which will be around 40 meters of depth. So we still have about 100 meters to go.
And now we're reaching 150 meters. It's going to get more and more exciting since we have more and more light down here.
Exactly. So we'll see the reef starting to become way more colorful. We can't guarantee anything, but we might see more fish. Oh yeah, that looks like a bottle brush in the back there. We'll zoom in on that, get a closer look. Yeah, that looks like a bottle brush black coral.
So it's the main front where it makes it.
You can see a sea spider in the back as well.
Yeah.
There was one that I saw while we were collecting one of the samples. It jumped off the back, just jumped off into the deep. Like that's going to be a long way down for it to fall. I think we don't really need to collect this. It'll be good. Thank you.
Yeah, we can move on. And now we just had a little discussion in our Slack group. So all the biologists are agreeing to move upwards to see what is on top. And while on our way to the top, we will collect rock samples at certain depth. I would also be very interested to see what kind of rocks are here, even though I have no idea about it, but the structure looks really amazing.
Yeah, exactly. I feel like we've actually seen this on a couple of the dives, these sort of caves and these crevices and the rock formation of we've moved upwards, and you know, it's such a complex habitat for so much to grow on. But yeah, I have to say I'm pretty much the same, you know, as we've said many times, we're no geologists, but you know, like a lot of the things that we've been discovering recently have really sparked our interest in geology and in rocks. So I think that's the great thing about being on a boat like this, is that even if you know, have one specialty, or even if you're passionate about something, you learn so much about so many different things that, you know, I've learned so much about jellies, about sea corals, but yeah.
Also just
Geology.
like understand coral reef in a historical way, how the reef is formed, not only because of the creature, but also how the tectonic plates are moving.
Exactly. And just looking at its geological history is so important and so fascinating, I guess, to understand how you get a different distribution of different animals.
These rocks are really crumbly.
Yeah. Can it cause more avalanches soon?
It's it doesn't have a lot of sediments on top, so
No,
yeah.
not as much as before. These orange soft coral, I think they are.
Gorgonians?
Or they look similar to the ones we collected last time, weren't they?
Oh, the yellow one, yes.
Yeah.
Then there is a chance that it might be black corals.
Yeah, it looks a bit more like the black corals because they're a bit more bushy looking, I guess, than the gorgonians that, you know, you can see the
Fan
grooves.
shape.
Yeah, it's a
Yeah.
lot more distinct.
Yeah.
And then on those, I guess you find, on the gorgonians, you find the pygmy seahorses, which are always nice to look through. We still haven't seen one in our time here, but
No, not yet. Now we're zooming into the rock to have a look at the structure. Marty is pretty happy with this sample that we just collected. I think like one of the things that I learned here is there's so many different ways for rock to form.
Yeah.
I never thought about that before. It's like rock is rock.
Exactly. As a non-geologist, you don't tend to think about it too much, but you know, already looking at all the samples that we have in the lab, you can already look through them and see how different they are. And, all you have to do is spark that interest, I think, in rocks, and then
Yeah.
it's quite easy to start to notice. You just need, you know, people like Rob and Marty to kind of teach you in the
Yeah,
beginning to
exactly.
appreciate the rocks.
We look at them shortly.
Someone actually asked, and this I'm curious about as well. How does Sebastian sense the altitude from the seafloor? I mean, I'm guessing
we
a
have-
sensor, but
We have beam that goes down and it bounces off, but it's way too high at the moment to register, so we don't know how far off we are from the seabed. The
Yeah.
distance. It's usually get to about 100 meters, so we know we're more than 100 meters off.
I don't know if you guys heard that, but yeah, the reason why it says, well, it doesn't have an altitude right now is because it's too deep to sense, and past 100 meters, usually the sensor won't.
So usually in another screen that we have in the control room, it will show the current depth, temperature, the timestamp, but also shows the pitch, the roll, the turns of the whole, the relative position of Sebastian, plus an altitude, and that's just a beam that they light, what's the thing, shoot down?
Yeah.
Yeah, beam that they shoot down to the bottom of the seafloor, but needs to be within certain range. Now we're really high up, so yeah, it's not in the range.
No, Jeremy,
And
you're breaking.
Exactly, Jeremy, because we would break the black coralometer. So,
Yeah.
you see,
yeah, it will be way out of the range.
exactly.
You might need to develop one of your own black coral meter and put it on.
I mean, there's no such thing as too many black corals, but I think it might be a bit hard to.
Sea cucumber coming into view.
Is a sea cucumber coming?
Where? Just on top.
In one of our other cameras, we've noticed a sea cucumber, so.
Yeah, but I was talking about
Big sea cucumber.
Yes, yeah. I'll get that one second.
Have to plot that black coral meter into a log scale. Sure. Oh,
Oh. Your favorite, Joan?
that's a pretty sea cucumber. This is I think it's the same one that we had previously, or at least similar one that had a worm on its body.
Oh, yeah.
Yeah,
The one that looked like the same, almost looked like a baby one.
yeah.
Do you have any fun facts about sea cucumbers or anything?
Usually, what I find it really interesting is if you can see a sea cucumber crawling or just staying in the shallow. A lot of divers will try to do one very stupid thing, but they keep trying that because they will stare at their butthole, try to find and see whether there's a little fish hiding inside. I know it's a bit creepy to do that actually.
Yes, that's
But-
actually very creepy.
Just an quick question. The temperature right now is 22 degrees.
Nice change in topic.
You just break so much of my esteem right now.
So back to the sea cucumber topic. Exactly, we have an audience saying it's pearlfish. So this little fish is not really parasitic, but is also not providing any benefits to sea cucumber. But sea cucumber is using its its body internal kennel to provide this place for this fish to hide inside so it doesn't get eaten by other predators. So it's pretty interesting, and people are trying to have a look at that. Is also because sea cucumber can breathe through their butthole, so their butthole will open and close and open and close, and that's why people are staring at that places trying to see whether there's a little fish head poking out or not.
What an invasion of privacy.
Exactly.
That is a fun fact. Thank you. I think everyone wanted to hear that.
Forget it.
Like, the coral colors are getting so much abundant right now.
Trying to zoom in into something that could be a hydroid. Almost looks like that same target species we were looking for, but yeah, could also just be a hydroid. Yeah, so Jeremy, if you're watching, the expert, is there anything
Yep.
exactly? We're trying to tempt him to come back with the black coralometer.
Looks like a
Aglaophenid?
and Jeremy is nothing here. Okay, we can
Pondering.
move on.
Steady.
Oh, okay. We have a look at here right now and enjoy the pretty columns here on the branches.
All the cool stuff.
Yeah, no, I think we're good. Back to the poor sea cucumber that we didn't
This
harass.
one might not be poor. We haven't really looked into it yet.
Yes, we're just we just used him to think about it.
Yeah. It is a fact that it's really hard to forget once you know it.
Yes, I know. I think you have corrupted my brain, Joan. So Joan, just out of curiosity, actually, what actually was the thing that made you want to get into marine biology?
That is a good question. I think we had similar background because we were studying biology before. I think whoever chose to study biology is always curious to know what is going on with in general every life form. Why it's existing, why it's behaving like that. And once I started diving, I felt like there was so much more on Earth to think about, and they all look very different from what we've known on Earth,
Yeah.
on land.
It really is like another world.
Yeah.
Yeah, I know that for me, I always had an idea I wanted to, but yeah, it's like probably through diving that really convinced me that that's where I felt, most comfortable. I think also one of the other things is I grew up going to Thailand quite a lot, and there was this one place called Coconut Island, I think it was called, that we used to go to every single year, and
Wow.
it was super easy to access the reef because you could just go from shore, you just go and snorkel,
Yep.
you get beautiful corals, you got to see loads of reef fish, and I think through that it made me fall in love with it. But as I grew older, I remember there was within 1 or 2 years I witnessed that entire reef die pretty
Yeah.
much.
It's very sad.
Yeah, after that, we never bothered really going back because there was nothing left to see. Because because they didn't really protect that site, people could go snorkel, stand on corals. Obviously with bleaching, it never recovered, and I think something like that really struck me when I was quite young. And yeah, I guess there's a bunch of reasons why both of us would want to get into studying this realm, but I know that's definitely something that really struck me hard and made me want to do something about it.
And thanks for John to telling us a little bit more information about pearlfish. So other than sea cucumbers, it also has other hosts, such like bivalves. So it's also getting its name from
Yeah.
pearl oysters. So actually some of this fish can just be trapped in those bivalves or use it as a host at the same time. What's that? Oh, that's just off.
Yeah, we're gonna zoom in on the red
Right.
whip right in front of us. Yeah, obviously in everyone's time.
Looks like a Hexa Coral so far.
Yeah. Yeah, an octocoral. So Viminella or Elicella?
I think we can move on when you feel like it.
Got a beautiful big crinoid.
Yeah. Did you see that too?
These look like gorgonians.
Yeah.
Fingers crossed for a
Yeah.
pygmy seahorse.
seahorses.
Yes, exactly.
Everybody's eye wide
Yeah,
open.
exactly. What crosses my mind though is, you know, often when you go diving, people do tell you you should try to minimize the flash because seahorses and especially pygmy
Yeah.
seahorses have very sensitive eyes. So I'm thinking, you know, with Sebastian's lights on it, whether we're just blinding them. Well, I hope not. I don't. I'm just, you know.
No.
I know that they, you know, you can use some flash on them, but they just ask you to limit it, so you wouldn't be blinding them immediately. But
Okay.
yeah,
Hi, Casey. The guys say hello.
The first orange soft coral we collected actually looked like this one. You remember in the depth, the orange one?
oh yeah. Mm-hmm. Is that a little critter on it? Or...
It's gonna it's a fair portrait. It's gonna be hard to
Yeah, no worries.
Yeah.
We can also move on.
I'll look at that.
Jeremy says it's the wrong coral for
That's how many pygmy seahorses he's seen.
Paragorgia? Question mark, maybe?
That's just my guess.
Paragorgia?
What's that?
I don't know.
I'm not an expert.
Do our experts know?
Yeah, do our experts know what type of coral is this one? We've got a guest in the control room saying, is it Paragorgia? And we also got a question from Jeremy. How many of pygmy seahorses have you seen before? Why are you so certain about this? It's not a coral for them? It's a little damselfish swimming under the shade.
$1 million. 200 inches.
I think they're just trying to move.
Yeah, I think we're trying to get into a bit shallower waters and seeing if we can sample anything there. I know, it's so tempting to stop for every single thing we see. Especially at these depths when everything looks like it could be something interesting.
Yeah, I don't even know where to put my eyes.
I know. That's why almost sometimes the deep, like really deep sea dives are almost easier because, you know, when we see one fish, everyone knows exactly what to look at, whereas here your eye is kind of looking everywhere.
Yeah, I think we had a Russian audience, but unfortunately, smile back to your smiley face, and I really don't know.
I don't think we have any Russians on board. Do we? No.
No.
that I am aware of.
Russians.
Ooh,
Hooray,
so we
finally
have a-
fish.
Finally
Okay.
fish. We have a Russian fish person.
Yes.
Rock, rock, rock.
Rock.
Right.
Oh, and more fish. Yeah, we really are starting to see more.
And there are more sponges here from another
Yeah.
camera here. It's,
Yeah, very big sponges.
yeah, it's coming. The sponges on top. It's really big. It's a really big sponge.
Yeah.
A few of them.
Which rock did you see?
I just looked at a ledge, cavey ledge or something, so. Yeah.
So we're trying to sample more rocks here from Marty as we reach a new depth.
This is the depth for the last glacial maximum.
The
Yeah, we definitely want a rock from around here. This is when sea levels were down at the last high surge,
Oh.
100 metres, 120 metres, and you get these overhangs coming to the rock. So we want to grab this.
So the reason we wanted to collect a rock sample at this depth is because last ice age, the sea level was 120, around 120 meters lower than current sea level. So by grabbing a piece of rock from the overhang here, we can analyze, try to interpret what was happening at that time.
yeah, we can also compare, I guess, our samples to the other samples that we collect.
Found the loop. Come again? Found the loop. We found the loop. Where was it? Was that little black bucket? So what's up there? Yeah, it was. Oh, that one.
Yeah, I love these formations. It's really crazy to think how they formed this. Almost seem like it's defying, not exactly, yeah, gravity in this case.
Gravity definitely played a very important factor here.
And it makes you think what's hidden inside these sort of
Yeah.
crevices and caves.
And now we had a watch change in the control room. Chris went to take a break, and J-Rod Cody is back with us for the ROV controlling.
There's plenty of little fish in there. It's really booming with life here. I just look at this and I think of all the things that we could sample.
We have way more than, no, we have way less ball boxes
Oh,
than we
yes.
expected.
Unfortunately.
If we wanna, yeah.
Drop that.
Yeah, so this rock here seems very crumbly, so we're trying to just get a big chunk out of it rather than a... And the exciting thing is when we collect these samples, or if we can collect a bigger rock sample than this, often Marty can pass over the biology samples that are on this rock, so
So they can
it's
be
almost a
analyzed.
win-win situation where we get more samples than we collect even. Okay, third time's the charm. There you can get. Yeah, it's a stubborn rock that just won't break, I guess. Someone just asked if we're at the top yet. So the top of this reef is at approximately 40 meters depth, so we're getting there soon, but not quite. And I think there's so much to explore around here that we're not really in a rush, I guess, to get to the top, because I think there's a lot to be done at this depth. Yeah, and as you can see on the map here, we've really had this steep ascent, which, yeah, sometimes I guess when you're looking at the live stream you don't realize how steep it is because the ROV is just going up a bit like an elevator. We get this steep wall that we're climbing up.
This rock is really stubborn.
Yeah, been working on it for a while. Doesn't even look like there's much to support it, but it goes to show. I guess that's good, with like a huge formation like this, you wouldn't want it to be more fragile or anything could collapse so quickly.
Especially thinking about all these detached reefs have been existed here longer than the barrier reef, so they must be very tough to sustain themselves in an open ocean with all the currents.
Yeah.
Without any protections.
Ramming speed.
Yeah, exactly, because when you look at the map, you can really see it kind of almost stands alone, as a sort of building. People said that it's, as tall as, was it the Eiffel Tower or the Empire State Building, which is crazy to think. We've just easily climbed that. Easily, I mean, obviously for the ROV pilots, they're the ones doing the ascent, but
Oops.
oh,
Trying to see if our stabilization arm can do something.
just push it out.
No.
It's really just stuck in there. Yeah, pretty hard rock.
It's been millions of years.
Temperature. So, someone just asked if the chemistry of the rock has any effect on the species that deposit themselves on. I mean, obviously, Marty, I think you can answer better than me. I know that a lot of species, need a hard surface, but could you comment, I guess, a little bit more on?
So was the question about whether the chemistry of the rock
Yes.
influences what biology?
Yes.
No idea.
Yeah, I mean, I would have thought a lot of things are just opportunistic and just attach onto anything,
Anything
they
that's
can.
hard.
Exactly.
Exactly. Yep.
But
I don't know.
who
Actually.
knows? I
Yeah.
think that's a good
It's a
PhD
good question.
project for
Yeah.
someone out there. Yeah.
Yeah, so the chemistry of the rock, of these rocks, is calcium carbonate with various minute quantities of other trace elements incorporated in there as well. So whether that influences the biology that encrusts on it, I really don't know.
Yeah, I think it would be very hard to study that.
Yeah.
So. I'm not quite sure if we're ever going to manage to get this one out of there.
We've kind of reached that cusp where we've
Yeah,
persevered. So
exactly.
it's
We have to
do we just keep
continue.
going for a little bit longer or let it go. I think
It's hard to let it go when you've put so much effort into it.
This will be the last chance to get a reef rock at this depth, and as you two both so eloquently explained earlier, this is the sort of, we're at the water depth now where during the last glacial maximum this would have been sea level, so shallow coral reefs would have been growing at that time, so that's why this particular depth that we're at now is really of interest to us.
So is that something that you keep in mind, I guess, for every single dive that we do to make sure that you sample that depth?
Yeah, so it is something we keep in mind on each dive. We kind of take into account prior knowledge and understanding of where we've seen various different terraces on throughout the mapping, not just on this expedition, but for the decades that our colleagues, Jodie Webster and Rob Beeman and others, have been studying these Great Barrier Reef shelf edge reefs. So we know that at certain levels we consistently see these terraces which represent past sea levels, and so we keep our eye out for those all the time for opportunities to sample, and then if we take samples from different geographic locations or different latitudes from similar depths, then we can make comparisons between them. Again, it's kind of all part of that process of trying to piece together the geological history,
Yeah, exactly.
and rates of reef growth, timing of when there were hiatuses where the reefs weren't growing.
I think that's what's so interesting about geology, it seems, is that you collect a rock and you almost have to see it as you're looking into its history, and it's a new story.
Exactly, yeah. So, one analogy or descriptive story that I've heard that really resonated with me was, if you imagine that you take a history book, a big thick many pages history book, and you lie it down flat on its side, and you take a hole punch and drill through it with a hole punch, and all you're left with is all that little confetti, and each
Yeah.
piece of confetti has a few letters on it of the history book,
Yeah.
and that's all you've got to go by. And from that, you've got to try and piece together the whole
Decipher.
book.
Oh, wow.
You know,
Really, yeah,
So,
it does kind
yeah.
of point out how hard a geologist's job is, I guess, to
so the more little hole punches you can collect, the more
exactly. The more
pieces
your
of the
story might
that
make
you
sense.
can try and put together.
Yeah, that is a great analogy.
Yeah.
Never thought about it that way, but
Yeah.
because I'm guessing also a lot of the time when you get a rock sample, obviously you have an idea of what explains, you know, different layers, but how often is it that you're 100% sure of, you know, how to describe the whole rock,
Yeah,
every single layer?
Well, it's so rare to get really good samples. As we're seeing here, it's really difficult. Great samples come from cores, from coring. We drill down through a section of reef from the surface down. It might be, you know, I mean, optimal would be, you know, 20 or 30 metres. Our colleague Jodie Webster has a core from the Ribbon Reef area in the Great Barrier Reef, which went down to 220 metres.
Oh, wow.
And so those cores have been logged. They've been studied over years through many different projects.
Okay. How do you study those cores, actually, if they're 200?
Oh, they're broken into sections.
Okay, and
So
then you
they're
just-
broken up into smaller, manageable sections.
Okay, yeah.
And they literally get laid out and used on the table, and you
You
start
just
at one
look
end
through.
and work your way through.
Wow.
So you log the community assemblages, so the different corals that are present, and not only the corals, but all the coralline algae, the forams, microbial lights, so limestone rocks that are precipitated by microbes. So you look at all those assemblages and associations and try and work out.
Big asteroid just above you. Go straight up.
Oh,
Oh yeah,
Go in.
nice.
Right in that crack there.
Yeah.
That's a sea star.
oh, that is a very big sea
Yeah,
star.
big fat sea star.
Cushion stars maybe, or something like that.
A very bulbous one.
Yeah.
So some genus, genera of coral and also coralline algae are quite good depth indicators. We know that particular species probably had about a 5 metre depth range. Optimally, that's the sorts of things that we're looking for. So then when we see those down a coral core, we can put together when was it shallow and when was it
Yeah.
deep.
Okay. And I think, yeah, that's what's so interesting about these cores that you mentioned before. Like, I know your PhD, you used a lot of data from that was collected a long time ago.
Yeah.
And I think with, you know, even just a few cores, there's so many questions that you can ask on one single sample.
Exactly.
And that's why you can reuse these samples. And
Yeah.
I think that's what's so great about it. And I think something that Rob pointed out the other day was, you know, what we're collecting right now is, you know, obviously for us and for our own discovery, but for future generations. And that's something
That's
that,
beautiful.
yeah, I love, yeah, wow.
Yeah, that's right. I mean, you really can't put a value on what we're collecting on this expedition. It's everything is super valuable to us, but then of course for decades to come and PhD students 20 years from now, or in my
Yeah.
case the cores I was working on were 35 years since they'd been collected.
Really.
So yeah, every sample is so valuable. So all of those curators out there that like to have big clean outs and throw things out.
Yeah. Think about the future
Yeah,
generations.
think about the future. Yeah.
Yeah, it's another reason why I guess there's pressure on us to make sure we label everything properly, and
Yes. Yes.
I know it's a big process that we have in the
Yes.
logging and in the lab as well. To make
Yeah,
sure.
I was really fortunate with the cores that I've been working on. They're 5 metre long vibra cores, they're called. The vibra coring is a particular method for coring through unconsolidated sediment. And when I located the cores at Geoscience Australia down in Canberra, and I was looking through the digital data that accompanied them, the metadata, there was even the original field notes from the
Oh,
scientists
really?
that collected those cores in the
Yeah.
field at that time. So they described how it felt when the vibrocora was going through the sediment and when they hit a
Yeah.
hard surface, and all of that description was all
Oh,
still
wow.
still there for me to grab hold
That's really
of.
interesting.
So, yeah.
Did you say that you met the collectors? who
Yes,
stayed
so
there?
the cores I worked on were collected by Professor Peter Davies, who was at the Bureau of Mineral Resources, which is now Geoscience Australia. They were collected on a survey in 1983,
Oh wow.
and Peter Davies was the supervisor of my PhD supervisor.
Okay.
So family tree,
Yeah.
and all of these collaborators are
Scientific
still in
family.
contact with one another and still work
Yeah.
together. And there were a couple of other PhD students at Geoscience Australia at the same time that were working on other cores different to mine, but that were also collected by Peter Davies.
Okay.
And so he came in on one of the days that we were down there logging the cores. So he came in and had a look at what we were looking at and chatted with us and
Oh,
talked through some of the samples. So yeah, that was really cool.
yeah. How great it is to be able to see people using some of the things that you collected,
Yeah, I can't
you know,
imagine
30 years
from
ago.
now
Exactly.
if someone's messaging me asking questions from my
Yeah.
PhD.
Or looking at the notes that we're logging right now
Yeah.
in our sea log. I know, obviously, because we take a rotation out of who logs in our sea log here. We all range in expertise, but sometimes we'll look at something and it'll pass by and we'll go, okay, is that a sponge? Is that a coral? And you'll log
we're looking
sponge
around
coral,
at each other.
question mark. So I know a lot of the notes are a bit cryptic sometimes. So yeah, I'm talking to the PhD student in 30 years, whoever looks through that. Apologies for,
And the spelling.
Yeah, and
Some of our
spelling,
on the
exactly.
scientific names. So, yeah, enough's good enough.
yeah.
I think we should probably start making our way towards the top. What do you think,
Yeah.
Rob?
What's that?
Yeah, I think we need to start making our way towards the top.
Yeah.
Yeah.
Be on the look for a rock.
Let's have another look for a pygmy seahorse and then make our way up there.
We need to, make our way up. The meso photic people are waiting for us to get to the top.
Yeah. Well, not a lot. They're rare. On these big fan. This is pretty.
This is really nice.
Yeah,
This-
lots of seeds when these soft corals.
Gargantuans or stuff like this as well?
I think. Hmm? Yes, I think one of our goals as well is to, have a look at the Gorgonian sea fans. Hopefully spot a pygmy seahorse.
Well, yeah, try hard. Yeah, okay. Alright, so we should move up.
I know that one of the big questions, I'd read a few, few blogs about it. I'm not sure if they ever knew the answer, so if anyone out there knows, I know on different pygmy seahorses, you get the texture of the seahorse that, matches pretty much exactly the coral, and a lot of people were wondering, if this is because, the parent seahorse as well, it matched, it was genetically determined, or whether the pygmy seahorse actually grew up learning to camouflage depending on the coral that it settled on.
Good question.
Yeah, that's something
have no idea.
I actually tried looking it up, and I read
did you find
several things. So I'm not sure if there is one definite answer. I think it was if they settled on it, that's what determined
Okay.
the texture. So it wasn't fully genetically determined, but partially, obviously. Yeah, I think that's because whenever you see one, they're always
Yeah,
exactly the same.
Not only colour but texture
Exactly,
as well.
the texture, and it's what if it decided to let go and find another one? I don't know how easy it is for them to find another fan, or if they really just stay on that one fan their whole life. For pygmies, anyway. I know that other seahorses can swim and find a new coral to settle on, but I think because the pygmy seahorses rely so much on that camouflage, I wouldn't be surprised if they try to stick onto that one coral for the rest of their life.
Okay. Well, as much as there's lots to look at here, we need to start our climb towards the summit. So the very top of this reef is in about 40 metres of water depth, and we really don't know what to expect, what we're going to find at
Yeah.
the top.
It's
Whether
always
it'll
so
be-
exciting.
Whether it'll be kind of barren
Yeah,
and wave
could
swept,
be.
or whether it will be a reef like
Yeah.
what we're seeing here.
Whether there's something waiting for us at the top, I'm always hoping.
There we go. That's a great shot. So that's the ROV SuBastian's lights have all been turned off, and this is the ambient light shining through the water coming from above. So quite a lot of light here. We're at 115 metres depth. And looking out to the side.
Yeah, it really is a great shot. You get that eerie.
Yes, and
Twilight
there's
Zone
the
feeling.
silhouette shot. Beautiful. Sometimes we get fish swimming around in the silhouette, which looks really awesome.
Yeah. It's a bit scary, actually.
Now this invokes sharks circling around at the top of
Yes.
this reef, doesn't it?
So this is the summit. It's just
Yeah, so the summit is ahead above us.
we're on the very eastern edge of it. Yeah, it kind of wraps this way.
A lot of sponges here. We're at the sponge and Gorgonian fan coral garden depth, aren't we?
Yeah, it's beautiful. Starting to get much more fish as well.
It's really fun stuff.
Some of these sponges get huge, don't they?
Yeah, they really do. I was just trying to look if I don't think the lasers are on, but yeah, there were a couple that look like I could fit my face in them.
Nice little fish there. Which I have no idea what it is. Maybe you do, or
Neither.
maybe someone watching can
No.
tell us what it is.
Yeah, if we have any fish experts out there. Yeah, I can't say I'm a fish expert. I know some, shallow coral reef fish. But especially at this depth, I wouldn't think I'd be able to recognize any of them.
There's a question on YouTube asking what plant life is there on shallow reef in the Great Barrier Reef, which is a good question. So I guess a lot of the plant life is algae. It can be macroalgae, and also seagrasses. You get seagrass meadows in the Great Barrier Reef lagoon. some algae is fleshy and some is coralline or has a carbonate skeleton. And that can be red algae, green algae, particularly the halimeda, which is the type that I've been studying. I've been looking at them from a sedimentological perspective because they have a calcium carbonate skeleton that produces a lot of sediment. But certainly in the Great Barrier Reef there's vast areas that are dominated by halimeda, calcareous green algae.
Yeah, listening to your Marty presented her PhD completion or exit seminar the other day, and we all got to listen to it, and yeah, it was really great because, you know, everyone obviously talks about the importance of the coral reefs and the Great Barrier Reef, but it's often the case when people slightly overlook the you know, either seagrass a little bit, but people are starting to talk about seagrass a bit more, but halimedas, like those kind of habitats people often don't really talk about, and their like, biological importance. Or
Certainly in this northern section, north of about 16 degrees latitude, which is about in line with Port Douglas, from there northward, the interreef part of the Great Barrier Reef lagoon, so not the coral reefs, but the seafloor, you know, around in between is dominated by halimeda. Produces a lot of sediment, a lot of calcium carbonate.
actually, this is a random question, but I guess one that I'm a bit more curious about considering the project that I'm doing. But I don't expect you to know the answer, but do you happen to know, because I focus on remote sensing, so I look at a lot of substrate types in terms of their reflectance.
Yeah.
Do you know how much different species of algae, so like halimeda and Chlorodesmus, there are a couple, how much they differ spectrally?
No, I don't know,
Okay.
but the answer is I don't know. But I would imagine that there would be some differences, well, certainly between fleshy macroalgae and calcareous algae. I would imagine that you would get different reflectance from, well, the hardness of them, but
because my project focuses on trying to differentiate between algae, sand, corals, but I'm trying to incorporate, I guess, the most comprehensive spectra for algae by trying to choose obviously different types of algae. So what I've done is I've picked a few species, because what I've read is I've seen have the raw data for these spectra, and they do seem to differ a little bit, not too much, but I don't know how significant it is. I don't know if it's just natural.
Yeah, just within natural variation.
Exactly, or if it really is species specific. That's why I just thought I would ask.
Well, yeah, you'd have to do the trials and see.
Yeah,
and
exactly.
test it.
I know, coral species often have a slightly different spectra, but also, even for my project, I've found like an average of different species, because that's what I know most people tend to do, and it's already very hard to differentiate between the different substrate classes, so, between algae and corals, so looking at species specific, it's even harder, and I know people do try to do it, but it's already hard with all the interspecies variation.
What about rugosity? Do you incorporate rugosity into your kind of calculations as well?
I haven't.
Activity and rugosity?
I haven't, but I've read a paper that has tried to take into account the texture.
Because that might also add like complementary.
And I think a lot of the time with the different textures and the 3D structure of coral habitats, you often get a problem of non-linear spectral mixing. So my project focuses on unmixing pixels or readings to see what proportion of different spectra would influence this reading. So if I have one pixel, I'll look at, does 80% of it consist of like a coral influence, and does 20% come from a
at
sand
the pixel level?
I mean, it's more of a point.
There we are looking upwards. We're at 80 metres now,
Yeah.
looking up. We haven't got too far to go before we get to the top of this
Yeah.
reef.
But to unmix the spectra, there are many different algorithms to do it, and
Are they bannerfish
yeah.
or are they Moorish idols?
but yeah, what I'm focusing is trying to unmix it in a linear fashion, whereas I know a lot of people say that this spectral mixing doesn't often occur linearly because of, say, a 3D structure of a coral reef. So you would expect a non-linear mixing because of, you know, the spectral interaction between the ground or between, you know, different substrates, and especially when you get, yeah, as I said, a 3D structure, it doesn't mix as linearly as you would expect.
And here we have some Halimeda starting to come in. So the green segments, this is a coralline algae, a calcareous green algae that we were talking about earlier when we were talking about what plants. So Halimeda is one of the most prolific sediment producers in reef ecosystems. And I can see a little bit of Leptocerus there.
Yeah.
Okay, Tom, we're starting to see some things that might be of interest to you now. Just have a look. So we've collected a few Leptocerus corals already for Dr. Tom Bridge. He's the coral curator at the Museum of Tropical Queensland. I know that he's online today watching the YouTube feed, and there's certainly in the depth range now that is of interest to Tom. So for us, we're getting really shallow now. For Tom, these are deep corals. Starting to get into the depths that will be of interest to people that have been waiting all day for us to get to the top.
Exactly. It's going to be a short portion of the dive, but
Yeah.
an exciting one.
So it looks very similar to things that we've collected on previous dives, but to me they all look very similar. So I'm probably not the right person to be talking about these corals.
Yeah.
And we can just it's not jumping up and down at all. I know that we are looking for Acropora in this area.
There really is so much going on.
There seems to be quite a bit of a delay between what we're seeing on the control room and the YouTube. So we'll just try not to zoom around too quickly, or our colleagues on shore will be lagging behind. We probably have, so Tom, just reaching out to you, we probably have half an hour or so if you wanted to join us on the commentary. We could organise that, or just let us know if you want to just watch on the YouTube feed at home. A few more fish now.
Yeah, plenty fish that I'm starting to recognize.
And there we go. That's the ambient light, the natural light coming through from above. Okay. So Tom won't be joining us on the commentary, but I guess we need to keep our eyes peeled for these aquapora corals, branching corals that you're looking for in these depths. So Jeremy, I hope you're watching too.
Always watching. Yeah, even when he's working in the lab. 'cause he has
Extracting
it in the bag.
DNA.
Yeah.
Certainly looking more reefy than Tideman Knoll, although the top of Tideman Knoll was a lot deeper than this.
Yes.
Much deeper, it's true.
I don't see any aquapora just yet, but it is a little bit like finding Wally, I guess, when you've got so
Yes.
much going on.
That's right. Oh, where do I look?
Where's Waldo?
Where's Wally?
Where's Wally? Is there a difference in between like UK and in the US? Okay. So in the US it's Waldo.
Waldo. Where's Waldo?
Okay. Yeah, I grew up with Where's Wally.
Me
so-
too. I grew up with Where's Wally.
Yeah.
Wally is the Australian Waldo. Weird.
Not weird, just different, depending on where in the world you are.
Yes, Diana, it is breathtaking, isn't it? We're certainly very privileged to be able to be here and see this unfolding before our very eyes. A lot more reef fishes coming in here.
Yeah, there's plenty.
Yeah.
All hanging around that soft coral.
I'm going to check a few of the platy corals. Plating, okay. Get closer to some of these plate corals. Yeah, so the top of this reef is in 40 metres, so if you were lucky enough to be able to come out here on a dive boat, you could just about dive on the top of this reef.
Actually, that's true. Could probably dive this part as well. 60 is a little bit deep, but
Too deep for me. I know there's plenty of people that do, but I
I think I've
don't
gone
have,
to
I
about,
don't have any training in technical diving or next
No,
case
neither. I
myself.
think the deepest I've gone is 50.
Okay.
Not further.
Yeah, deepest I've gone is 45, just on air.
But even there, you know, you spend a couple minutes and then
Yep.
you have to go back.
Okay.
So
So Tom, if you're watching, we're going to just take a moment here to have a look at some of these plate corals. And zoom in a little bit. He hasn't picked out any particular one, but he'd like to get in a bit closer. Just have a look around.
there's one up there. The dark one.
Sounds amazing.
they really look like corals, especially there.
Sure.
Yeah,
Layering over like
yeah.
that.
I mean, those weird pink things look like sponges, but
Yeah. So for everyone watching at home, we've got a internal chat on Slack here between ourselves and the scientists working with us that are onshore, and so we're just they're messaging us and we're trying to communicate back to them about what we're looking at, and of course there's a slight delay between us and them, so just I don't think we'll be doing sample collecting of any of these, but just zooming in to get a good look to identify what they are. Leptoceros, do you want to look at the other ones, the smooth plates? Okay. Yeah, these ones. You want to collect one of these, Tom? Okay, we can try. We'll try. I'm sure they'll be very delicate and breakable. And I'm guessing, Tom, it's not you're not targeting anyone in particular, just something that looks like it's reasonable to try and pick one up and collect. So thinking it might be Montipora. And
it's got an interesting
Tom,
texture.
yeah, can you give us an indication, Tom, whether you want a smaller one where you get the whole thing, or are we just breaking off a piece of one of these bigger ones?
Yeah.
Okay. All right, we'll do our best to try and get a piece of this one.
Looks like it'll just snap right off.
Yeah, I think it'll be pretty brittle.
Yeah. We just
There we go.
perfect.
Perfect. And I think we'll just put it in 3/4 at the back. Please. Yep, and some bonus halometer on there as well.
Is there anything that you could do with it, Marty?
No, other than admire it, no.
You've probably seen enough Halibut.
Yeah, I was gonna say you're probably sick of Halameda by now. Are you planning on doing more work with Halameda in the future?
Yeah, I think at some point in the future. I mean, there's certainly plenty more work to do. If anything, you know, the PhD that I've been doing for the last nearly 4 years has answered some questions. It's partially answered some questions and has raised a lot of new questions. So there's certainly more work to do, more work to be done. Thank you very much.
Someone else, please, sir.
No, I think that's fine. Okay. So Tom's saying that these Montipora are, I think they're in the family Acroporidae, and although they're fairly common in the mesophotic Great Barrier Reef, we still don't know specifically what it is at the species level for that one. He says, thanks very much, that's a great sign.
So it's a Montiporas. It's interesting, streamer-like sponges.
Yeah. So we've probably got about 20 minutes or so left on the dive, so we'll just continue doing what we're doing and just browsing around. fairly, yeah, just keep taking your time. Closely.
Hey Marty,
Hey,
back at the desk.
so good to have you back, Rob.
Take us, go to the end here. It's been quite a dive, hasn't it? We've climbed to the mountaintop.
Yes, which is sometimes we don't always get there for whatever reason.
Yeah,
At a
yeah.
time, or it gets getting too rough, or for whatever reason.
But we have
Last
climbed
couple of
this
dives we've done
full
well.
500 metre tall mountain, and lots of coral. You know, that was a big question mark. You know, we knew it was about 40 metres at the top. Was there coral there? And we got it plainly in front of us here. Lots of soft corals and sponges and halimeda and
Yeah.
lots of coral.
And interestingly, so you and I this morning were asking ourselves, what would it be like? What would we see on the top? And would it be compared to this dive or that dive? And one of the things we brought up was how would it compare to Cairns Seamount? And that which was on previous voyage. And Tom's commented here, completely different community to Cairns Seamount.
Okay,
Yeah.
all right. We've got Trevally, a
we
couple of
are.
Trevally in the background.
We're at the top.
Perhaps. Happy to be corrected. It's maybe a big whip black coral. Yes. But look how these plate corals are all stacked out, all pointing up towards the light.
Yep. Broad and flat surface to maximise their surface area for capturing that sunlight.
Yeah. So maybe just a bit of a recap about what we've done here. This leg 2 of the northern depths of the Great Barrier Reef. We've been at sea for 3 weeks now for leg 2. Your leg 1 was a week before that, so you've been at sea now for nearly a month.
That's right.
And the voyage will continue on until the 17th of November. But this is a mushroom coral.
Coral.
Yeah, so we've dived as often as we could through this period. A lot of mapping, as many dives as we could squeeze in. We even got a bonus trip up to Torres Strait.
I don't know my fishes.
Yeah, we had to go and refuel because we just travelled so far in these last 2 legs. You know, thousands and thousands of kilometres here, including all the mapping, which happens 24/7 when we're not diving.
That's right. When we're not diving, we're mapping 24/7 through the night, mowing the lawn, as we call it, up and down. And that's been, well, for me and for you, I think it's been one of the highlights. Is getting up in the morning and seeing what was mapped.
That's right. Yeah, it's like a Christmas present. What have the guys done
How did it
overnight? And you know, there you have the ocean revealed in, you know, beautiful 3D, and of course we can colour it and shade it how we like because, you know, it's all digital. But then the big question is what's living down there, and so this is the beauty of the Sebastian. We get the 3D models of the seafloor, and then we can say, all right, we plan our dives. And I think every dive has been very, very interesting. I am super pleased that there's lots of coral at the top of this 500 metre tall coral reef, which is really an ancient reef, but only the living
It's a very thin veneer on top.
thin
isn't it?
veneer,
Very
yeah.
thin veneer.
In
It's
this
living coral reef.
top photic zone where the water's warmer and there's enough light. Yeah, a lot more fish happening up here, isn't
Size of
there?
that plate coral.
Lots of fish. Whoa, okay, that's busy. Okay, start
Rightio,
counting your fish.
people. Come on.
I'm seeing surgeonfish and damsels, anthias, lots of damsels, very busy.
Alright, Chris.
Fantastic.
Hello? Yeah, that's it. You can kill it.
A little Moorish idol at the back. You know, the yellow and black stripe, really obvious.
Wow, it's busy here.
Lots of damsels, butterfly fish, wrasses. See some wrasses in the background. It's been the sponges, Marty. I've really noticed the sponges on this dive.
Yeah.
Those big vase sponges down on the steeper walls, really obvious. And of course, all in amongst all this shallow coral here, all of this
All
kind of
these branching
spindly.
things.
Yeah.
They're sponges.
They're all sponges, yeah. And we got such a variety here. Big favids. Saw Tom was saying Montipora. Yeah, really, really interesting. Somewhere. Oh, big cod.
Here we go.
Yeah, this is a big coral trout. A big trout of some kind. Yeah. Someone would get rid of the fish, they're obstructing the view of the coral. Yeah. Only Jeremy would say that. Yeah, but it's all coming to an end. Tomorrow is our last dive, and that is the last dive for this expedition, and very likely for the remainder of the year. There won't be any more ROV Sebastian dives here because the ROV team is having a rest. There's a big crew changeover in Cairns at the end of the week. So we will have to, bid farewell to all people watching ashore. It's been an amazing ride, hasn't it, Marty?
Yeah, it absolutely has. Still plenty more to come, although tomorrow is the last ROV dive.
That's right. And so for leg 3, there will be no ROV diving. It'll just be mapping and some other. We got some interesting things planned, like visiting these very remote islands out in the Coral Sea Marine Park. So we'll continue doing social media, but unfortunately won't be through.
So Jeremy, we've just seen your message about
Sebastian.
focusing on the branching soft corals, but we probably have passed by where you exactly spotted those. So we'll just slowly look around, and when you see something of interest, you'll need to jump on and let us know. Branching soft corals is what it is.
Yeah.
Shark.
Shark in the background. We're going to spin around because we've got
There it is.
There it is.
In front of us.
Oh, what's it? A grey reef. I think this is a grey reef shark with a very dark caudal fin. Yeah, a little bit itchy. You watch it. So he brings his pectoral fins down and does his very tight turns. This is very cool, Marty. What a great reef, eh? We've got
There's a couple of them around. Yeah.
oh, here it comes. Oh. That's an in-your-face shark.
Beautiful animal.
Yeah.
Well, if we said, let's imagine what's at the top, and we said, and sharks circling around. Here we have it.
Fantastic. The shiny texture on the skin. It seems to be agitated. There's another one in the background. Another shark coming in, smaller. 2 sharks.
One grey, one white.
White
Okay.
tip, is the other one coming in.
Yeah. We got the shark song going. Lot of fish. These are surgeon fish. These black ones down the bottom. Big school. Okay, the shark's not going anywhere. Yes, so tomorrow's dive, we want to go out with a bit of a bang. Not literally, of course, but we are going to take Sebastian down to 2 kilometres down into a plunge pool that is at the very entrance to Wreck Bay. And Wreck Bay is just north of us here. Quite a spectacular bay when mapped in 3D. Quite interesting. We've actually dived on the northern arm of it. That was at Wishbone Reef.
A unicorn fish there. So that was quite a few days ago now that we did that dive, given that we've had the refuelling expedition in between. But we'll come back to Wreck Bay for our dive tomorrow.
Some Moorish idols down the bottom. There's 3 together.
Back to the corals.
Yeah, to the corals. Enough fish, enough sharks. Yeah, righto, Jeremy. Oh, it's busy down there, isn't it?
Tom.
Yeah, Tom. Oh, lovely. These are nice anthias. I think these are anthias.
Here we go. So is this one of the branching soft corals that you were referring to, Jeremy?
So we're still talking to our colleagues ashore, but just talking a bit about tomorrow's dive. Oh, there's that shark again. So we're down to 2 kilometres deep. We wanted something sort of topographically very spectacular, and we found one of these deep plunge pools. But we also promised Dougal Lindsay at Jamstech a mid-water dive, and so we're going to try and combine 2 together. We'll see if we can get Dougal on for about 3 hours of a mid-water dive into the water column, and then we'll have to call it quits, and then we'll dive down to 2 kilometres deep and just see what we can find at the entrance to Wreck Bay.
Yeah, we won't be able to linger. It will take us a long time to get down and a long time to get back up.
Yeah, that's right. So we're looking forward to it. I love these steep and deep dives, and we'll have a chance tomorrow.
Look at this one.
So we hope there'll be a bit of diversity with, you know, the mid-water plankton dives, which we really enjoy, with Dougal's excellent commentary.
Yeah. They're good fun, aren't they?
Yeah.
Dougal is so excited.
Yeah. And then we'll take down 2 kilometres, and I really enjoy the deep water environment. The cold water corals, it's just an utterly alien, foreign
Really
environment.
otherworldly, literally, isn't it?
Yeah.
Okay, keep looking around for a 3D branching coral. A 3D branching coral, so branching in 3 dimensions. Branching this way, plane. That's what he's looking for.
Is that what, soft coral? Or
Soft coral, I think.
soft
he's
coral,
talking about.
right? Yeah. Only a halometer.
Yeah.
What's that? Square pot antheas. Radio.
The shark's still swinging around.
Yeah.
There's a tussle going on here between sharks and corals.
Oh, there it is. Oh, wow.
Beautiful.
Yeah, that's a silvertip.
2 of them.
Silvertip sharks. We saw grey.
2.
Okay, we're getting quite a bit of shark action happening here. So we've seen silvertips, grey reefs, and we saw a white-tip reef shark earlier in the dive. Oh, wow. Fantastic.
Well, we haven't seen this on any other dive.
Not this
Saw
many.
We saw the 2 that were behind us the other day.
Yeah, a bit of a glimpse, but this is terrific.
Not like this. Every dive's been really quite different on this leg. I mean, we're trying to plan for a bit of diversity, but they really have, each dive's been really quite different. Because when we're planning the dives, of course, we don't know what's down here, we don't know what we're going to see.
Yeah, yeah. So we make some educated guesses. I always start with what's the most diverse topography that you can get in as short a horizontal distance, and then of course we have to bring in environmental conditions and wind and waves. But here we've been lucky. Today is just flat, calm. It's like a swimming pool outside. Been really lucky with the weather.
Yeah. Yeah, we're coming back.
Yeah, so tomorrow down at 2 kilometres, we're probably going to be looking at about 4 or 5 degrees Celsius when we get right down. So it really is cold. Just to give you an idea, when we bring the bio boxes up from those depths, and when you plunge your hand into the boxes which are filled with water when it's back on deck to take out the samples, you really can't leave your hand in very long. It's like putting your hand into ice cream. It's just really chilly and it starts hurting. So that's the cold temperature down there. It's Antarctic intermediate water that's flowed up from the Southern Ocean.
And he wants to keep looking down at the corals. Exactly. Gee, some of these plates are huge, aren't they?
Yeah, they are like, tabletops.
So is that for Tom? Because I've got Tom here getting instructions as well.
Oh, he said go on top, right? I thought we just saw that sojourner just.
current there. That little
Yeah.
one just keeps swimming. You can see the sun just blowing around a bit in the current as well. Okay. Well,
Yeah,
so
there's so-
we'll keep looking around.
Yeah, we won't probably stay too much longer here because we're getting near the end of the day. It's a squirrelfish. Like the top of this newly discovered reef is about 300 meters long. There's our sharks. And about 50 meters wide. So it's now 100% mapped, and we can. It's not a danger to navigation, but it's certainly a really spectacular reef. To think that its base is 500 meters lower is pretty amazing.
Yeah.
I've said this a few times today. It's not every day you find a 500 meter tall reef in the Great Barrier Reef. So for us, it's been a real privilege to mountain climb this newly discovered reef, the full 500 meters, and find that there's quite a thriving coral sponge community at the top. Oh, an anemone, big anemone here. That is just the kind of anemone that you have an anemone fish. And there's
And there we
clouds. Fantastic.
There we are.
There we
It's a
go.
great highlight to it.
Yeah, there we go. Let's just linger here a little bit. So the biggest one will be the female, and the others are sort of the males. But it's
This is classic, iconic Great Barrier Reef.
yeah,
right
it
here,
is.
isn't
Yeah,
it?
yeah. This is the
That's right. It's the first time we've seen anemone fish in an anemone. on
yeah,
these
yeah,
dives.
yeah. I wonder if this is a spine cheek. Can the fish people tell me? Are we looking at spine cheek anemone fish here? Just having a bit of a zoom. There we go. Just poking their heads out.
Beautiful, aren't they?
Amphiprion Akindinos.
Fine cheek.
Thanks, Joe. Not spine cheek. Okay. Yeah, you're right. I can't see a spine. We just need to zoom in. Yeah. Beautiful. And when you scuba dive on the reef, they're pretty common, aren't
Yeah,
they?
yeah.
You see them a lot.
Absolutely.
All different kinds, different
Yeah,
kinds
that's
of
right.
anemone
Yeah.
fish.
We think of Nemo, but they don't all look like Nemo.
That's
There's lots
right.
of
Well,
different
that's your
ones.
classic
Yeah.
clownfish. But these are all anemone fish, and they're all obligate, associated with these anemones. Oh, is it the
going
baby? Oh no, I was just thought it might have been a baby. They have a different coloration. It's just a little,
Oh, this little one.
tiny little one on the left. Oh, what's that? Is it? It's tiny. It's just flipping around a little bit. Okay, fish people, what do we got here? It's got little stripes on it with a very dark tail. Very active.
flamboyant.
Yeah, sort of drifting a bit like a leaf, I think.
Quick.
Yeah, it's gone. There we go.
Maybe a cleaner wrasse of some sort, yeah.
A Labrador's wrasse, yeah, maybe just a very small one, juvenile.
Frame crabs. Vicky, did you get a few frame crabs and clownfish?
Very nice. Oh, what have we got? A big surgeon, a unicorn? No, what are these called? Lunate? A lunate surgeonfish, is that it? And we got unicorns.
Fish.
And just behind it, the one behind it, it's a.
Wow, we've got everything here.
Yeah.
Unicorns.
That's right. We just need a few pink
Need a
unicorns.
few rainbows and unicorns.
Yeah.
Sharks,
Sharks.
clownfish.
Yeah. Just need a stingray to go past.
A turtle, that's what we need.
Yeah.
We need a turtle.
Yeah. Oh, that's nice. That's it. That little fish there.
Yeah. Yeah.
It's a Labrador, I think, a little wrasse. Baby
one suggestion
juvenile.
is a little cleaner wrasse. Juvenile cleaner wrasse.
Yeah, it's busy. There's just the sponge variety here. Like these tall ones, that strange vast, like one.
Yeah, the iconic fish shot where he was in the enemy and just his face popped. It was an awesome shot.
Great pose.
Word was, dude.
He's very photogenic, isn't he? He or she.
Yeah, Glenn's asking questions about some of our sensors. Yeah, for sure. I mean, we're logging all of the oceanographic climate data, so temperature, salinity. This is happening all through the dives and also at the vessel itself. And when we do the 3D modelling of the terrain, we use artificial light. It's the same sort of tools that are found in game technology, 3D game technology. Is that wrasse again?
Little wrasse.
Okay, fish people, what do we got here?
Type of wrasse, and this might be juvenile cleaner wrasse. And you can clean a wrasse and clean other fish.
That's interesting, the background. It's a strange looking fish. I haven't seen anything like that before. Very odd. Is it a damsel? Got really odd coloration. Spots? And an orange head?
I wonder if it's a juvenile of something else. You know how
Yes.
juveniles often have different colouring.
Yeah.
So,
Yeah.
the old is really recognisable, but when you see the juvenile, they can be quite
Yeah,
different.
yeah. Nice Anthea coming in.
Beautiful. What a treat.
The
Finished
best
today.
fish action we've had
It really
all
is, isn't it?
this whole voyage.
Gemma Galbraith, I hope you're watching.
Mm.
Maybe you'll catch up on the recorded video later.
Yeah, Gemma was with us on the last voyage. She was a fish enthusiast. It was great having her in the seat here when we're in these shallow waters.
Juvenile Bodianthus.
Yeah, okay.
Bodianthus and Theoides. Yeah.
Oh, yep. Juvenile, yep. Yeah, so tomorrow's dive, we'll probably, we're starting about 9 each day. It'll be a big day, though. We don't usually start any earlier. It starts, people have to wake up and get themselves prepared. Oh, big coral trout. Very healthy.
Wow. I wonder what age that one would be.
Hopefully like a big female, lots of
Hopefully,
eggs.
Yeah.
Oh, what's that?
Hello.
Oh, trumpet fish.
Trumpet fish.
There you go.
We've got everything.
Just quietly sitting there.
Open up the shallow coral reef textbook and
Yeah.
start reading them out. We've got them all.
Yeah. This is good. Yeah.
Flute mouth.
Flute mouth. Yeah.
Okay.
Really interesting animal. Like the jaws on it. You can see how they open up. Yeah, mean.
Yeah, he does look mean.
A very predatory. When they decide to swim, they do this flick, and they go from 0 to 60 in a fraction of a second.
Wow.
Fistularia, flute mouth. Yeah, you can see they get that tail, they just give that a flick and boom, they're gone. Oh, a bit of a wander. What have we got? It's just after 4 here. How much longer can you guys handle? All right, maybe 10 minutes or so. Let's just go for a wander.
49. Slow creep up to 40 metres.
Yeah, we're at 49. We measured it was around 40 metres, but we took
The
tide
into
off it.
account,
Yeah.
So, yeah, it could be 43-ish.
There's that
There's
big
that
cod.
coral trout. Put the lights on. Oh, okay.
Some Moorish idols. Xanthus cornutus. See?
Is that the Moorish idol?
Yeah. I do know some fish species names. Ah, okay, Kai, good. Orange and white fish, Bodianthus and Anthioides, the
Yeah,
lyre
yeah.
tail hogfish. Yep, thanks to Kai. Kai was he's been part of all of these 3 expeditions, going right back to April, May. We had Kai join us for the Visioning of the Coral Sea expedition. That was great. He brought his fish army on board because at that stage we had no fish people involved, and now it's expanded into this huge collaboration around the world. It's been really interesting to see the fish people get involved and take the imagery and start developing scientific papers around it, because that's really what I guess this is where the work really starts for us. It's great looking at this vision now, but we have to take this vision and actually do some science with it, and that means counting, measuring, describing, and really trying to make sense of all the trends that we're seeing here in the deep Great Barrier Reef and Coral Sea. So we had a meeting just a few days ago with Brendan Brook and everyone from Geoscience Australia, just talking about the last expedition. Huge amount of data and imagery collected, including from an autonomous underwater vehicle that was taken out to sea, owned by the Australian Centre for Field Robotics. So all of that imagery and vision is now being worked on by people all around Australia. And I think it's going to keep us busy for some time, isn't it, Marty?
Many projects and sub-projects that come out of that work.
Got a big school of damsels in front of us. Some more anthias.
The size of some of these plague corals is just amazing.
Yes, I'm thinking, given it's just a little bit deeper than your typical shallow reef, perhaps it's just enough to be below wave base to really smash it up, because we do know that in that high wave energy area in the really, really shallow waters, like the top 5 meters, it can be almost bulldozed. This is a pretty healthy cover of living coral here.
Yeah, the diversity and the cover is pretty high, isn't it?
Yeah.
Alright, Chris. This goes off.
Oh, what's this? This is a red sponge. Yeah, this is as deep as I've ever dived, Marty, and I've been diving for decades. 45 metres as deep as I ever
Yeah,
get.
45 is
Usually I'm
max
at
depth.
20. I never go much deeper than 20 metres.
Often you're around 10, 12 for much of the time.
Most of the
as
colour,
well.
yeah. But I did a deepish dive on Osprey Reef, which is sort of southeast of where we are now, with a boat called Undersea Explorer.
Oh, yes. My 45 metre dive was on a wreck in Port Vila in Vanuatu. Yeah, he said, can we look at this coral? But which coral is it? This one? Oh, this one? Yeah. That.
Which one, Chris? Roger.
Oh, wait, so to the right, so I'm not. Just let him catch up because there's a lag between us and him. So, Jeremy, this purple one or the one to the right of this one? The one to the right. To the right? With the branches going straight up. Yeah, I think he means, yeah.
Yeah, the water temperature here is about 26 degrees C.
That one. No, not that one.
Be very comfortable diving here.
Yeah, that one.
No, I'm just trying not to run over these other pools.
Some algae in the background.
Yeah. Yeah.
What's that?
That's cool. That's coral. Okay, so Jeremy said we can pass on that one. So I think we might just about need to call it.
Yes, we've got a lot of mapping to do overnight. We've got only 2 more nights left here, so given it is the dive today and also the last one tomorrow, that really only leaves the night time for doing any of the mapping remaining in this far northern Great Barrier Reef. So really at the very sharp end of this leg. So what we might do is just do a slow pan backwards and say goodbye to everybody ashore who's been following us through the day in this really spectacular.
It's really been a
500 metre
fabulous dive, hasn't it?
yeah.
With a really spectacular ending. So thanks everybody on shore for joining us and playing along at home.
Yeah,
It's great having you there.
yeah, and thanks for the rest of the team, all the ROV team, Chris, Cody and J-Rod and James and John on the Ycast and the team here, Marty McNeil, myself, Rob Beeman, got Valerie Cornett, Joan Lee and Vicky Lowe. This is the science and ROV team and the rest of Falkor saying farewell and hope to see you tomorrow morning on our final dive for the SuBastian down to 2,000 metres.